Homeostasis Ray here. Welcome to Unit 3 Cell Energy. We'll be starting off with Lesson 1 under Cell Transport and Homeostasis. Now, today we're going to be talking and learning about how cells move things in and out of the cell, for the various things that they need. We're going to use that knowledge, then let us learn about photosynthesis and cell respiration, which deal with energy and how it's either stored and then later used or gotten from an outside source and then used for cells to be able to power themselves. So let's get started on cell transport and homeostasis. Turn to page 2 in your note packet. Unit 3, Lesson 1, Cell Transport and Homeostasis. So in class today, we worked on organelle spotlight, where we spotlighted 5 organelles that you learned about in middle school, and we wanted to bring them back into focus so that we could apply them to our knowledge in Unit 3. In Lesson 1, there are 2 specific ones of those 5 that we have to really focus on: cell membrane and cell wall. You can take a look at your organelle spotlight if you need to know more about these, but the main thing is that cell membrane, every cell has it. It lets things in and out of the cell, but not everything in and out of the cell. So it's why we call it semi-permeable. A cell wall is permeable, which means everything can go in and out because that's on its main job. Its main job is to provide structure and function. And what we're going to find out is that plant cells in particular are known for having cell walls because they need them because of how much water they have to bring into the cell. So we're going to start with our notes, which is passive transport and tonicity. When we think of the word passive versus the other word aggressive or active transport in this case, we're going to associate passive with not having any energy. There's no effort. It just happens naturally. So I want you to write that into your notes, that this happens naturally, that there's no effort, no energy needed. So let's define it. It's the movement through the membrane with a concentration gradient from high to low. And what that means is that it is a whole lot easier for things that are moving across membranes to go from someplace that's really, really packed together in high concentration and strong, and then leave to go to a lower concentration. In other words, move to free space. We see this happening when we cook. These air particles that have smells attached to them are the highest concentration in the kitchen or the nearest thing to the pot. And as we move further and further away or over time, the smell travels through and becomes more and more dissipated. This is actually diffusion that is happening with air. And we'll talk about what diffusion is next slide. So this movement is going to continue this process of moving from high to low concentration until there's balance. And we call this word equilibrium. And again, passive transport is specifically known for being natural, no energy needed, zero energy needed. There are 2 types of passive transport: diffusion and facilitated diffusion. With diffusion, this is still going from high to low. We see this with gas, with liquids, pretty much any kind of molecules that are moving from high to low, it's diffusion. And when we apply it specifically to the movement of water from high to low, we call this osmosis. So again, osmosis is a type of diffusion, but it's specific to water. So if you see the word water in your quiz or someplace else, you know automatically osmosis is what's going on. Facilitated diffusion is diffusion that needs a little bit of help, because to facilitate means to help. And so what's happening is that it's still moving from high to low, because that's what diffusion does, but it's involving these big molecules, and so it needs this help of these proteins to move the molecules from high to low. So when we look at these 2 images, which you're going to have to do on your quiz and identify which one is which, if you see the proteins and it's going from high to low, facilitated diffusion. No protein, but still going high to low, it's just regular diffusion. Let's talk about tonicity. So we have to define first some words. Solution is 2 or more substances mixed evenly, usually in a liquid state. In biology, we will be speaking specifically about solutions that are dealing with water, nothing else. Chemistry, you'll find out that their solutions are not always in water. But in this case, we will be dealing with water. And there's 2 parts of this solution. We have the solute, which could be anything from like sugar or salt or anything that's dissolved in that water. And the solvent, in this case, water. And let's define what tonicity is. So this is the concentration of the solution. I want you to underline solution in your notes. which determines the direction or extent of diffusion. We're talking about diffusion, specifically osmosis, because we're dealing with water again. And what it's saying is that we look at what is surrounding the cell, and the liquid that's surrounding the cell, we look at the number of solutes in it. And if it is high, we call this hypertonic, which is coming to our next thing, hypertonic. When the outside solution contains a higher concentration of particles, so water moves outside the cell. This means that we have all these molecules outside and less molecules inside the cell, so water is then pulled out of the cell and it causes the cell to shrink, just like in the images to the right. You'll notice that the bottom image, although the interior part of the cell membrane is shrinking, the overall structure of the cell, the line in black, is not, because that is the cell wall. And this is one of the things that plants need, because plants need to maintain their shape. And the cell wall allows them to do that when dealing with the movement of water in and out of their cells. Let's talk about hypotonic. Now, this is when the outside solution contains a lower concentration of particles, so the water moves inside the cell instead. So outside is low, inside is high. To help balance, more water is pulled into the cell and it swells up. In the case of animal cells, it actually even can burst if it swells too much. Plant cells, because they need a lot of water, the cell wall protects its shape. Now, think about the first part of this word, hypo, and the last word, hyper. I want you to write in your notes, think hypo low, hyper high, and that's how you're going to try to remember the difference between the tonicities. The last part is isotonic. This is when water moves into and out of the cell to maintain balance. Because iso meaning same, and think isosceles triangle. And in this case, as one water molecule moves in, another water molecule moves out. And this is happening because the solutes, the materials inside and out of the cell, are at equal balance. And this is showing all of them together. Now, in your notes, we have this chart. And we need to know when it's hypertonic, that means outside of the solution is high, it pulls water out, so it shrinks. This goes the same for the plant cell. And in our notes, we want to write, this is happening because the H2O, the water, is moving out of the cell. With isotonic, water is moving in and out because the amount of solutes inside and outside of the cell are the same. So it's moving in and out of the cell. Hypotonic, that means what's outside of the cell is low, so the water is pulled in, is going to swell because water is moving inside the cell. And the cell structure differences is why whether a cell bursts when it swells up or not. And the main thing is animal cells don't have cell walls, so they don't have that structure, that protection that keeps the cell from bursting like plant cells. Now, I'm going to do this first one together, so watch how I fill this out. I find the black dots, those are the solutes, and the red dots with sort of white sort of sides, that represents the water molecules. And I have word banks and result to cell word banks that I can also use. I also will be drawing arrows. So the first thing I do is I count how many black dots are inside, which I had 4, and how many dots are outside, 2. When I look at those numbers, 2 and 4, outside, which is the only one I care about, is the low number. Because it's the low number, that means that my water is going to be rushing into the cell to balance the high number of solutes that are there. So that means all of my water molecules are going to rush in. And if they all rush in, that means that this solution is going to be hypotonic and it's going to swell. So you're going to write that in your notes. Make sure you add that part to your notes. You are going to do the other 2. So we've already done hypotonic. So that means the other 2 are either going to be hypertonic or isotonic. And again, use your word banks and also draw your arrows. Part 2, active transport. So just like we talked about passive transport where it's natural, there's no energy needed. If you are active, you are definitely needing energy. And we're going to define active transport as the movement through the membrane against the concentration gradient from low to high. When you write against, maybe off to the side, think shove. If I'm shoving something into something, I'm having to use my muscles. I'm having to use energy to move those muscles. And again, we're going from low concentration where we're all spread out and then we're packing things in. Think about the effort and the uncomfortableness that it's going to take if you had to pack a bunch of people into an elevator. Once you get very close to high capacity, you may have to shove and move and hold, and it's going to take more energy to sort of hold yourself in place than if you all had room to spread out. And again, active transport is needing energy because it cannot do it because, again, it is having to push things back in where it's not naturally supposed to flow. And there are two types that we're going to talk about. We have the protein pump, which this is proteins that are used to work against the concentration gradient. Again, it's against that word. We're going to go from low to high. But the main thing is that it uses proteins. So this is another type of transport that uses proteins. The difference is when you look at the image, you're going to see the word ATP. In your notes, write ATP is equal to energy. It's the type of energy that our cells need. So it has that energy. If you see ATP in an image and then you see a protein, you know it's got to be a protein pump. The protein actually pumps the molecule through. The next type is bulk transport. So this is the membrane-bound vesicles to transport materials inside and outside of the cells, again, going from low to high concentration. You'll notice that if you see groups of clumps of molecules, it's going to be bulk transport. And bulk means in high concentration. It's groups. You might even want to write that in your notes. And there's two types, depending on whether it's going into the cell or going out of the cell. So exocytosis, think exit, is going to be leaving the cell. So you'll notice in the image, the arrows, it's inside the cell when it starts, and the arrows are showing that it's being fused with the cell membrane and then being released. Versus endocytosis is the exact opposite. It's starting from outside the cell. It is then forming as vesicle and being taken into in a group. Think internal, into, endo. Here are some more practice. You'll use your word bank to then go on and fill this out, which you can do on your own if you know what you're doing and read your notes. The last part of our notes. We have passive and active transport help maintain the body's internal balance. So this is really important. This is why your body has this happen to all of their cells. Homeostasis is really important to understand. And we define homeostasis as the state of steady internal physical and chemical conditions maintained by living systems. This is actually something that living things, it's a characteristic of living things that they maintain homeostasis, balance. In the human body, just in one example of tons, thousands of examples of homeostasis in our body is how we maintain a regular temperature. When you get too cold, you start to shiver. When you get too hot, you sweat. And it's the way of the body to either cool you down or to heat you up. When you shiver, you're actually contracting muscles, and that is going to release heat, which that is going to warm you up. Versus if you're sweating, you're releasing liquid onto your skin to help cool that external or internal layer of skin down. Some other examples of what we do is regulating water levels in our body, maintaining calcium levels in our body, and maintaining a healthy blood pressure. All of this is homeostasis at work. Now, this is the very last practice. It's actually the easiest practice, in my opinion. All you're doing is matching and understanding whether do any of these need energy or not. So I'll help you do the first couple. So exocytosis. Exocytosis is a type of active transport, taking things, exiting the cell with it. So it's going to need energy. Diffusion is a type of passive transport. It's not going to need energy. Number 3 is just a picture. But when I look at that picture, I'm going from high concentration to low concentration, and I have a protein. So that means that's facilitated diffusion, which also means no energy. You are going to do 4 through 10 on your own. Good luck. Make sure that you're doing these practices on your own before you get to class, and start studying early. Have a great day.